moving on to the ST or like Visium data here that I wanted to just give a bit more information about. Like I said, there is some confusion here regarding the terms spatial transcriptomics, ST, and Visium. So I just wanted to give a bit of a backstory to this. And the saga sort of begins here in 2016 when the Fresén and Lundeberg lab and a lot of other people as well published this paper in Science by Ståhl et al., where they presented a technique that was called spatial transcriptomics, and in short, ST. Which sounded like a stroke of genius, like to claim this like whole field as the name of your technique. But once you have 40 different other groups that also do spatial transcriptomics, it can become a bit confusing, and it usually is, especially during conferences. When they ask which sort of spatial transcriptomics method you do, and you say, I do spatial transcriptomics, and you have a discussion back and forth. But the concept here was to use spatially barcoded arrays to capture mRNA. And some other groups use the technique as well, but between this time period of mid-2016 to late 2018, it was mainly used by like Lundeberg and Fresén lab, some other people as well as that, but the heavy users were definitely here at SciLifeLab. However, in late 2018, 10x Genomics acquired the IP rights to this technique, and in, wrong direction. In late 2019, they launched this Visium Spatial Gene Expression Platform, and to somehow be able to separate Visium from ST, because Visium is extremely similar to ST. It operates at a higher resolution, has some different reagents and stuff, but the concept is more or less identical. We now tend to refer to data from this old platform as ST, legacy ST, original ST, ST1K, and sometimes Visium, and that's mainly by very unattentive readers of manuscripts and even reviewers, and that's of course incorrect. We also have some prospects of what's going to happen here with the Visium HD platform being launched in late 2022, at least according to the JP Morgan presentation that 10x had like a week ago. Let's see. And I think it's mostly relevant for us to speak about this Visium platform. That's the type of data that you will be looking at. So I wanted to give you some specs just so you know what you are actually working with. So this is an array-based technique. It has a 6.5 millimeters times 6.5 millimeter area that you put your tissue sample on. In this area, you have about 5,000 spots, 4,992, arranged in a hexagonal grid. And the spots are 55 micrometers in diameter, and the center-to-center distance is 100 micrometers. Each spot has millions of capture probes, and that's about as specific as I can get, because 10x won't really disclose how many millions are. But they also have this, each probe has a spatial barcode, which allows you to associate transcripts back to a spatial position. They have a poly-T capture sequence, which allows you to capture the mRNA. And Visium more or less then operates depending on what you define as the full transcriptome at the full transcriptome level. You can't really yet distinguish between isoforms and so forth, but yeah, that's more of a technical definition question. And as we said before, it's about 1 to 10 cells that contribute to each spot, differs depending on which tissue you're looking at, and yeah, how many cells you have at the exact same spot, how cell dense your tissue is, but that's a good estimate, I would say. And I just want to emphasize here, it is not single cell resolution. That's a common misconception that I hear quite a lot, where you call cells, spots cells, for example. And I should also emphasize here that you get an H&E image, an actual image of the same tissue that you get spatial transcriptomics data from. And that might seem pretty relevant if you're only interested in the transcriptome data, but it's an extremely valuable reference resource. I spoke with people in the group that actually developed the SlideSeq data, and they seem sometimes to experience a bit of frustration to not have this like reference image to see what if it makes sense or not. So getting an H image is very valuable in most cases, and pathologists tend to really like it as well. And just to show you an example here of what the data could look like, in the very leftmost picture, we have these spots arranged, or like you see the spots under the tissue. In the second image, you see this overlaid on this H&E image that I was speaking of. You really see how it like fits well to the outline of the tissue. And then of course, since you have transcriptome data for basically every gene in this tissue, you can use to visualize the expression. And I did this here by letting the phase color intensity be proportional to the gene expression value. And this is breast cancer tissue. And as you can see, ERBB2, that's a breast cancer related gene. So it should be really well aligned with the cell dense regions in this tissue. And it seems to be. So So a word on this distribution then. As you perhaps have discussed previously, single cell data is usually modeled as an over-dispersed Poisson distribution or negative binomial alternative gamma Poisson distribution. Those are different names for this. And this is sort of important because that serves as a basis for several analysis methods such as normalization, differential linear expression analysis, and so forth. And at least according to me, I would say that this assumption is applicable to ST and Visium data. And we had to actually look a bit deeper into this in the stereoscope publication because there we use a negative binomial model. And what we did then to assess whether this was a fair assumption or not to make was to cluster this Visium data sample here and sort of said that, well, this is a very rough approximation that each and every one of these data points in the same cluster, we can sort of think of them as coming from the same distribution. We plotted the expression values like as a distribution here and tried to fit different parametrical distributions, such as the negative binomial, the normal distribution, and the Poisson distribution to these densities here. And in every case, actually, the negative binomial gave the best fit, even when you controlled for it having 2 parameters compared to the Poisson, who only has 1. And there we used the BIC and AIC criterion. So at least, and this is my opinion, but apparently went through with the reviewers as well, that using an NB distribution is fairly okay when you work with both ST and Visium data. Right, so for some brief words on the analysis of ST and Visium data. I should say here that we have tended to see quite a lot of batch effects between sections that are even from the same tissue specimen sometimes. So it's good to try to account for this, that you're working with spots from different sections in your data if you have several replicates, for example. And to account for these effects, single cell methods have worked really great so far, for example, like harmony and a C transform from CRAT. And then it's also good to remember that the cell density is often not really homogeneous across the tissue. So it's good sometimes to normalize your data based on the library size to account for this. And most normalization methods already do this, but it's just something that's good to keep in mind in the back of your head, sort of. And like we've discussed for some of these methods, and that is applicable to ST and Visium, the expression profiles are actually mixtures. And sometimes it actually makes more sense than to analyze them accordingly, looking at factor contributions perhaps rather than hard clusters. You could of course do hard clustering, but just keep in mind then that your cluster might not really be a cell type. For the single cell mapping, this is often improved, I should say, by using highly variable genes or a curated list of genes that includes, for example, marker genes of the cell types that you try to map. You usually do get pretty decent results, at least with the stereoscope method that we used, if you just take like the 5,000 top expressed genes, but it depends a bit on how distinct your different cell types are. So this can really improve your results. And I also want to say here that trajectory inference is really tricky when it comes to ST and Visium data. So far, I haven't really seen a good method that accounts for this fact that we could have several temporal states being present in the same spot. So if we have cells that are at different time points in the pseudo time, but they donate transcripts to the same spot. And also this incorporation of spatial information has been done sort of like, yeah, pretty heuristically so far. So I'm yet to see a good trajectory inference algorithm for ST and Visium data. When it then comes to filtering and data processing, this is often something that people ask quite a lot about, and it's so hard to say like this is the magic recipe that will always work. It's very dependent on your data and also your question. But filtering of ribosomal, mitochondrial, and HP genes usually have a bit of a positive effect on the result, since these genes tend to like constitute a pretty irrelevant source of variation, again, depending on your questions, of course. And then there's something that maybe is more of a technical thing or experimental thing, where we have observed a bit of leakage around the edges of the tissue in our Visium samples. And we are not super sure why, and it's not a huge like issue, but we are very sure that diffusion within the tissue is very minimal, but near the borders, it seems like sometimes like transcripts have a tendency to leak a bit, and that could just be good to keep in mind. And I believe you have this included in the labs later on as well, where you can see this.